Hello everyone, I'm Prakarsh, a research option MS student at Mechanical and Artificial Intelligence Lab. Our goal here is to bridge the gap between machine learning and biological and clinical research. We are currently working on multiple projects which involve application of machine learning in healthcare and biological science. The first project I will discuss has more fundamental biology application. Here, we are developing machine learning based models to design novel antibodies for neutralizing the coronavirus. Our motivation here was to use the widely available proteomics data, like protein structures, for understanding the interactions between virus and antibodies to predict the sequence of antibody which can potentially neutralize a given virus. We've been using the information contained in protein structures of viruses and the corresponding antibody to train a machine learning model. The central idea behind this is that we have an antibody shown in blue, which has interactions with the amino acids of the virus protein shown in green. We can learn these interactions that occur between these two and make predictions of interactions that would occur between the coronavirus proteins and our proposed antibody. The same methodology can be used for predicting antibody sequences for neutralizing the virus in a new viral outbreak. One of the key components of this project is the isolation of significant features from the protein sequence data. After a series of feature extraction techniques, we trained the ML model on these features. This allows us to greatly reduce the antibody sequence search space and predict antibody sequences. Finally, for a given sequence of new coronavirus, we predict the corresponding antibody which has a high confidence score for neutralization potential and assess its stability through molecular simulations. We have also extended this project to use generative models to understand the virus protein sequences and generate synthetic protein sequences from them. This can help us explore the potential mutations that can happen in a viral genome. We train this generator on the data we have collected for the virus proteins and then ask it to generate sequences specific to the virus species. To validate these sequences for their biological feasibility, we have also developed a novel graphical protein autoencoder. This allows us to incorporate a strong sense of biology to our predictions based on the collected data. Such a model can be used to explore the mutation space of virus, and an exhaustive search can hint at the next potential viral outbreak. The next project I want to discuss has significant clinical basis, which involves a blend of computer vision and artificial intelligence. Here, we are developing an image processing toolbox for high throughput analysis of video data. This project is in collaboration with Rango's Research Center at UPMC. The central idea here is to study the motion of cilia on cell surface. Cilia are hair-like appendages on cell surface which are used to clear out foreign particles. The abnormal function of cilia has been associated with diseases like chronic obstructive pulmonary disease. Here is the video of normally functioning cilia. Notice the synchronous motion of cilia, which is very important for their function. The frequency of their beating, the length of cilia are all correlated with their healthy function. This vigorous and synchronous motion is what we are interested in extracting from these videos. Here is the video of abnormal cilia function. Both the videos have been recorded at the same frame rate, but it is a little tough to see the motion of cilia in this video because it is so slow and asynchronous. This is one of the features that we are interested in extracting from these videos. Such abnormal motion of cilia can be caused by individual events like smoking and anesthesia, or it can be caused by genetic defects, which cause malformed cilia associated proteins. The goal at our lab is to develop tools to extract these features associated with cilia motility from the videos. The features are like cilia length, beating frequency, and percentage ciliation of cell surface. To calculate these features, we use techniques like optical flow, which I've represented on this slide. The color intensity denotes vigorous motion. At time t1, where intensity is low, it shows that there is less motion. At time t2, we observe that there is comparatively more motion. Another interesting challenge here is to design an algorithm for automated detection of such cilia clusters from the video. Finally, we assess the obtained results for their clinical significance for enabling the clinicians to form a better informed diagnosis. We are also developing a deep learning based model to classify these videos into disease state and normal state. The goal here is to have a video input and use a deep learning model to extract the relevant features for forming a diagnosis based on the cilia video input. Such a model needs to learn the temporal dependence in video data and uniquely identify the ciliated portion of cells. Such goals give this project immense clinical significance and increase its applicability. A well-functioning pipeline for this processing task would enable clinicians to form accurate diagnosis quickly. Lastly, we're developing a complex neural network architecture for predicting the arterial blood pressure from other monitored vitals like ECG. This project is in collaboration with the Rogers Group at Northwestern University. Abnormal blood pressure is a risk factor for many diseases, and high blood pressure can add a lot of cost to medical expenses. Thus, monitoring blood pressure is very important for effective treatments. Our goal here is to predict the systolic and diastolic blood pressure values from data like ECG, PPG, and SCG. Here is a glimpse of the kind of data that we use to predict the blood pressure. ECG, PPG, and SCG are fed as raw waveforms. The machine learning model then learns to extract the important features from this data, like the time difference in peaks, synchronization of the waves, and such. This is then used to predict the corresponding blood pressure values, which is the systolic blood pressure and the diastolic blood pressure. We can see that the model is able to use the input data to regress to the systolic and diastolic blood pressure values. We expect that a more complex model would be able to perform much better on such data. Since the signals themselves may not be sufficient for learning the dependence between inputs and the blood pressure values, we use data augmentation techniques to improve model performance. We use novel signal processing techniques to convert one-dimensional signal into a three-dimensional input by folding the input signal over itself like a snake to form a two-dimensional signal, just like an image. Then we stack multiple such images together to form a 3D video input. This is then used as input data to train the ML model. The ML architecture we use is specially designed for this task to understand the temporal dependence in data through LSTM. LSTM is able to learn the significant features in time series data and also forget irrelevant features. At end, we use a three-dimensional convolution to regress to the desired values of systolic and diastolic blood pressure. We then compare our predictions with actual blood pressure data that has been recorded for the same patient, thereby validating our results. A well-trained machine learning-based model can potentially eliminate the need for conventional blood pressure recording equipment. All these projects have significant translation potential and involve designing and implementation of novel machine learning and data processing techniques. If you would like to know more about these projects, feel free to reach out to Amir or to me via email. Thank you.